Welcome to the problem-solving set on the review for DNA structure and replication. Question 1 states to draw a bidirectional replication fork as found in eukaryotic DNA replication. Label the origin of replication, leading and lagging strands, and the polarity of all strands. Okay, so a bidirectional replication fork means that there's going to be a replication bubble. So we have one strand of DNA. We'll draw the replication bubble in the middle. And then let's draw its complementary strand. Okay, so the first thing it wants us to label is the origin of replication. Now remember that the origin is in the middle of the replication bubble. So this is the origin of replication. Okay, now I'm just going to assign polarity to these original strands so that we can work from there. So let's say that the top strand is going from 5' to 3', and the lower is going from 3' to 5'. Okay, so now what they mean by bidirectional replication, that means that replication is happening to the right, but it's also happening to the left. So let's do to the right first. When we're looking to the right side of the origin of replication, remember that this strand is going from 5' to 3'. Now DNA polymerase creates strands from 5' to 3', and they read the template strand from 3' to 5'. So when the template strand here is going from 5' to 3' in the direction of replication, that means that this is going to be the template for the lagging strand. Okay? So we're going to draw Okazaki fragments. And all of these fragments are 5' to 3', going to the left. And remember that this is the lagging strand because the elongation of these strands has to happen toward the left, right? Because it has to happen in the 5' to 3' direction. However, the opening of the DNA helix in this side of the replication bubble is happening toward the right. So this lags behind because it can only synthesize as much as is opened already. Okay, and then for the leading strand here, remember that down here, the strand is going from 3' to 5'. So the leading strand is going to go from 5' to 3' as all new strands are, but this is just going to be one continuous synthesized strand that is 5' to 3'. So we're going to label this one's leading, this one's lagging. Okay, and this is on the right side of the replication fork. Let's look at the left side of the replication fork. So we're going to write in the polarities. And now let's do the leading and lagging strands. So here the lagging strand, remember that all new strands are synthesized in 5' to 3' direction, and the template for the lagging strand is in that 5' to 3' direction. So now the lagging strand is going to be synthesized at the bottom. Alright? And all of these new strands are 5' to 3', 5 to 3. Okay, and this is lagging. And now on this side of the replication fork, the leading strand is going to be on top. And it's going to be continuous 5' to 3'. Okay? So as you can see, when it's bidirectional replication, the leading and the lagging strands actually switch sides depending on which way you're going, because if you notice, the replication on the left side of the fork is going toward the left, where replication on the right side is going toward the right. Like that's how the DNA helix is being opened. But the strands polarities, the original strands polarities, are always staying the same, so it's going to flip. Question 2. If the replication fork starts at the right of the DNA sample shown below and moves toward the left, what is the sequence of the DNA synthesized as the leading strand? So now, replication is going to be happening toward the left, and remember that new DNA strands are always made in the 5' to 3' direction. and DNA polymerase reads in the 3' to 5' direction. So this strand above is going to be the template strand for the leading strand. Template for the leading. But now they want the sequence of their actual leading strand. Now because DNA replication is just finding the complementation, the strand below is actually already the complement of it. So if we want to write it in 5' to 3' way, you just copy it, so you start from here. So you say it's A, G, A, T, G, A, T, A, C, C, 3'. So remember, if they give you a double-stranded already, that it's already complementary. So when you find the template, you can just copy the other one and know that that's what's going to be synthesized as the leading strand. Problem 3. The diagram below depicts a unidirectional replication fork. In the boxes, indicate whether the end is a 3' or 5' end. So these two boxes on the right are actually talking about the newly synthesized strands. Now remember that all DNA strands are synthesized in the 5' to 3' direction. And if this is a replication fork, that means that replication is happening to the left. And you know that because the origin of replication must be on the right, and as it's going, replication is going along, the DNA helix is being opened. So if replication is going toward the left, that means that this strand is 5' because it is the leading strand, and all strands are made from the 5' to 3' direction. So if replication is going to the left, that means that elongation here is going to the left, and that's 5'. So that means that above, this must be 3', and in that same strand, the other end must be 5'. So that means, even though I don't have a box down here, let's write that this strand is 3' and ends in 5', which means that the box left is 3'. And that makes sense because Okazaki fragments, they're made from 5' to 3', just as all new synthesis DNA strands are made, but they're lagging because as the DNA double helix is opened up here, they can only go as far in and then synthesize a new strand this way until they reach the old Okazaki fragment. So those are the polarities. Now it says label the leading and lagging strands. So this one is leading as there's only one fragment. And these here are the lagging strand. And even though they're fragments now, later on, after DNA replication has been done, DNA ligase will come and join these fragments together. Now it says, describe briefly why the lagging strand is synthesized differently than the leading strand. Again, this is due to the property of DNA polymerase that only allows it to synthesize DNA strands in the 5' to 3' direction. So it can only synthesize 5' to 3'. And that's because they can only add new nucleotides to the 3' hydroxyl, and that's why synthesis can only occur this way. So nucleotides can only be added to the free 3' hydroxyl. Okay, so that's why they're different, but evolutionarily there has been enzymes that have evolved that can deal with this. So we have DNA ligase that comes after and joins all these small Okazaki fragments together into one long newly synthesized DNA strand. Question 4. A DNA sample has 48% G and C content. What are the proportions of the 4 different nucleotides? So this is just a different question that is an application of Chargaff's rules. Now remember, these rules are about complementary base pairing, where the number of A's equal the number of T's because they pair together, and the number of G's equals the number of C because they pair together. So if you have 40% G and C content, that means that half of 40%, or 24, must be the proportion of each. So G equals 24% and C equals 24%. And also, out of 100%, that means that there's 52% of A's and T's, which again have to be equal in number. So when you divide that by 2, you get that A is 26% and T is 26%. So these are the proportions of the 4 different nucleotides. And remember, this is only true if this is a double-stranded DNA, which we're assuming that this is. Question 5. Describe the forces responsible for keeping double-stranded DNA together as a stable molecule that stores genetic material. So there are 2 different types of bonds that keep double-stranded DNA together in the helix. You have covalent bonds and you have hydrogen bonds. So covalent bonds are the bonds that hold together each linear strand, so they bind together the bases, the sugar, and the phosphate group. Bases. And there's a special type of covalent bond called the phosphodiester bond. And that bonds the sugar-phosphate backbone of DNA. And then hydrogen bonds are between the bases when the 2 strands of DNA come together through complementary base pairing. So these are through base pairing. And remember that A and T paired together have 2 hydrogen bonds. And G and C together have 3 hydrogen bonds. So overall, these are the forces that are responsible for keeping double-stranded DNA together, and it makes it less reactive so that it's a stable molecule to store genetic material. Question 6. Content analysis of a certain plant species genome reveals 55% G content. Why does this result indicate a problem with laboratory instrument? So because this is a plant, that's definitely a eukaryotic organism, so we're assuming that a plant's genome is double-stranded DNA. Now in double-stranded DNA, the proportion of G has to equal the proportion of C because of complementary base pairing. However, in this case, the analysis shows that 55% of the genome is G. So if G is equal to C, that means that C also has to equal 55%. But if you add that up, that's more than 100, which obviously isn't possible. So this is definitely a problem with laboratory instrument. If it said that G is 50%, it may be possible. I mean, it's kind of unlikely that an entire genome would just be G's and C's, but it is possible. So the reason that this indicates a problem is because it is more than 50%, which means that the total content is more than 100. Problem 7. Carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur are the most common elements found in living organisms. Which of these elements are not found in DNA, and which of these elements are not found in the sugar-phosphate backbone of DNA? So what is DNA? DNA includes a sugar. It's a 5-carbon sugar, which is called deoxyribose. And that includes carbon and oxygen, and also hydrogen. It has a phosphate group, which has phosphorus, and it has a nitrogenous base, which contains nitrogen. So if you look, we have carbon, hydrogen, oxygen, all in deoxyribose. Nitrogen can be found in the nitrogenous base, and phosphorus can be found in the phosphate group. So the only element that's not found at all in DNA is sulfur. And remember in the Hershey and Chase experiment, that's why they used radioactively labeled sulfur in order to label the proteins and not the DNA. For part B, it says which of these elements is or are not found in the sugar-phosphate backbone of DNA. Well, we know sulfur isn't found at DNA at all, and in the sugar phosphate backbone, you don't see any nitrogen. And that's because the nitrogen is found in the nitrogenous space, which is the A, G, C, and T, and that doesn't include the sugar phosphate backbone of DNA. So I figured I'd give Professor Schmidt a break with the true-false, and I thought I would run through these quickly with you myself. So number 1, the requirements for genetic material include the abilities to store and express information, replicate itself, and allow for variation. Now these are the 3 things are all true. And that is why DNA is the perfect genetic material, because it stores the information, it allows expression through transcription of the genes and then translation of the gene. It also can replicate itself through DNA replication, and it allows for variation because there's different bases, and it could also go through mutations. 2. Chromosomes are about 75% DNA and 25% protein. This is false. Chromosomal composition is actually roughly 50% DNA and 50% protein. Okay. 3. The variation between different nucleotides is in the phosphate group. Now this is false. Remember, I'm going to write that down here. Every nucleotide has the sugar deoxyribose, and they all have the same phosphate group. The difference lies in the nitrogenous base. So remember, these can be adenine A, thymine T, cytosine C, or guanine G. So this is where the variation comes from. So basically, the sugar phosphate backbone of DNA is all the same, and the variation comes in the center of the DNA helix, which has the bases. 4. Adenine and guanine are purines having 2 rings each. Now this is true. So the purines look something like this. They have 2 rings, so these are purines. And the pyrimidines which are T and C, just have 1 ring. Okay? So that is true. 5. In a double helix, there are 20 base pairs per helical turn. This is false. And it's just simply memorization that each turn has 10 base pairs per helical turn, or 10 nucleotides. Number 6. 2 single strands of DNA are held together by phosphodiester bonds. Now 6 is another false. The backbone of DNA, which has the sugar and the phosphate group, that is held together by phosphodiester bonds. However, 2 single strands of DNA are held together by hydrogen bonds. So let me write that down for 6. To make the statement true, it would be held together by hydrogen bonds. And just to kind of drill it in your brain, remember that A and T have 2 hydrogen bonds between them. G and C have 3. Number 7. Twists in the DNA helix create minor and major grooves. That is true. And you may not learn about it in genetics, but in other courses, you can see that protein interactions with DNA happen usually in the major grooves. And number 8, DNA replication can only proceed in the 3' to 5' direction. This is false. Remember that DNA replication can only proceed in the 5' to 3' direction. So, just to clarify that, because that seems a little ambiguous, we have DNA polymerase, which is the enzyme responsible for DNA replication. DNA polymerase can only read template strands, so it reads templates in the 3' to 5' direction, and it synthesizes new strands in the 5' to 3' direction. And remember, this strict polarity rules is what makes a difference between the lagging and the leading strand in DNA replication. So this is probably the most important thing from this chapter, because teachers may expect you to forget that, and in the questions the polarities really matter in some answers. Okay, so this concludes the entire chapter for DNA structure and replication, and I hope it was helpful.